Fantastic, cool. I'm just going to go ahead and start. Courtney, if you can watch the chat and let me know and stop me if things are going on here. I wanted to just frame this morning's sessions a little bit and explain what our thought process is in putting this together. COVID vaccine work and COVID vaccine issues are definitely on the forefront of everyone's thinking. It's sort of every day in clinic, this has been an issue. And I think that, I know that some of the clinics have had conversations about this a little bit, but not everyone has. Want to acknowledge that Hillary and Becca came forward with a real desire to want to talk about addressing vaccine uptake hesitancy among patients. And so they're planning the second sort of half to 2/3 of the session will be a lot of interactive stuff about using motivational interviewing techniques and thinking about addressing hesitancy with COVID vaccines. So she's going to talk a lot about that. To frame it a little bit, I thought that I'd do a quick hits updates on vaccines related to COVID. And the idea here is that she's going to talk, they're going to talk a lot about how it's not about the knowledge issues, that there's a lot of emotion, there's a lot of sort of affective issues when it comes to acceptance of vaccines. But I do want to make sure that we're on the same page about the knowledge about the vaccines and what the vaccines are all about. And so I'm going to sort of run through a lot of things very quickly. The objectives of my part of it this morning, I'm going to spend a little bit of time just sort of making sure everyone's on the same page, understanding what are the mechanisms for the different SARS-CoV-2 vaccines that are out there, what are the mechanisms of some of the ones that are being proposed, so that everyone has a pretty good understanding of how they're built and what they're made up of. I want to spend also then a little bit of time being able to define and more importantly, being able to accurately describe what the efficacy of a vaccine means when you talk about vaccine efficacy to your patients. And then we'll spend time talking about the efficacy and pros and cons. And I want to spend time talking about the Johnson & Johnson vaccine. I think that everyone probably has a decent understanding or a working understanding of the mRNA vaccines. The Johnson & Johnson vaccine's only been approved in an emergency use authorization for the last week or two, and there's probably a lot less that all of us know about that vaccine. And so I do want to share the preliminary data and what the FDA used to make their decisions. And then also talk a little bit, touch on some upcoming vaccines that are likely to be brought to the FDA over the next few weeks or a couple of months. And then finally end with some late breaking updates. And when I say late breaking updates, I mean within the last 2 weeks, some papers that have come out regarding vaccines, the AstraZeneca vaccine, information about asymptomatic spread. And so those are the objectives of what we'll talk about this morning. So it's a lot. Mechanisms of the vaccines. I'm gonna run through it a little bit more in detail with the mRNA vaccines, and then we'll just sort of quickly go through the other forms of vaccines because a lot of them have a lot of similarities. And so the Pfizer and the Moderna vaccine, the ones that we're all familiar with, mRNA-based vaccinations. My brother-in-law, who is married to a woman from Germany, always wants to point out to us that it's really BioNTech that developed it and Pfizer's just putting it all out on the market. So, but I tend to call it the Pfizer and the Moderna ones. So those are the ones that we're using. The graphics from these descriptions all come from the New York Times, and I'll have links at the end. But if you really want to have a nice visual to share with your patients, the graphics from the New York Times really nicely show you how these are built. And so as a reminder, the SARS-CoV-2 virus, single-stranded, positive sense encapsulated RNA virus. Within that capsule, you have the spikes that are coming out, and those spikes are the ones that bind to the ACE2 receptor on cells, and then will meld in, and then that RNA then gets encodes the proteins to build the next virus. And so for the mRNA vaccines, what Pfizer and Moderna have done is they've taken the chunk of the mRNA, the RNA part of the gene that encodes the spikes, and then have taken that and encapsulated it in a lipid nanoparticle. And important to all this discussion is understanding that RNA, if you just inject the RNA directly into the body, it quickly degrades within moments. And so you need the lipid nanoparticles in order to get it safely to the cell. And for all practical purposes, the differences between the Pfizer and the Moderna vaccine, they're very, very minor differences, but they essentially take the same chunk of the RNA for both their vaccines. The lipid nanoparticles are the differences between the two and why they aren't the exact same vaccine, but it's the lipid part of it that's the main difference. And so then the vaccine particles then fuse with the cells. That mRNA is translated into the spike protein. The spike protein is the spikes of the cell of the virus. The spikes are a trimer, so the three spike proteins combine together and then form the spikes on the surface. And so once it's in body cells, those are produced and are presented to antigen presenting cells, and then your remarkable T cells, helper T cells and B cells do their job and produce the antibodies and start to recognize the spikes. And so when you have an invasion of your body of SARS-CoV-2, then those antibodies obviously are floating around, bind to and hopefully neutralize the SARS-CoV-2 virus, and or if they're attacked within a cell, then the attacked cell produces the spikes, and then the helper T cells come along and kill those cells. So all of that that's the mRNA vaccine. Shifting, so then the next category of vaccines that we have are the adenovirus vector vaccines. And I already mentioned to you that the, so those are the Oxford-AstraZeneca vaccine and the Johnson & Johnson. The Johnson & Johnson, sometimes you'll see it, the Janssen vaccine or the Johnson & Johnson. Janssen's a subsidiary of Johnson & Johnson, so you'll see them interchanged in the literature. And then the last one that I've put in there, I'm not going to talk much about, but the Sputnik V vaccine, the Russian vaccine, is essentially an adenovirus vector vaccine. And so what they've done in that case is they've taken the RNA segment for, again, the spike protein. They've reverse engineered it to a DNA segment. And the reason that they choose DNA, among other reasons, is because DNA is much more stable than messenger RNA and sticks around better. And that's part of the reason why it can be at higher temperatures than the mRNA vaccines. And then they stick it into an adenovirus. And in order to be taken up into the cells, the adenovirus has to stimulate the immune response, but at the same time, you have to make sure that the machinery for replication is deactivated. And so that's been done. Then the adenovirus fuses with the injected person cells, and then it does its thing, goes into the nucleus, and then produces the mRNA, and the mRNA then encodes for the spike proteins, and all the rest of the steps are the same. The Oxford-AstraZeneca vaccine uses a chimpanzee adenovirus. And so the chimpanzee adenovirus, because it's a strange virus to the body, invokes an immune response. And the Johnson & Johnson one has taken a replication incompetent adenovirus 26, and has used that instead. And the Johnson & Johnson adenovirus 26 has previously been used by Johnson & Johnson when they produced an Ebola virus that they've been testing. And other companies have used the same adenovirus, including the Russian version, which uses the same adenovirus, and then their second shot, they use a different adenovirus for their vector. I can't see people, so please feel free, if you have questions, unmute yourself or let me know. I'm going to touch on the Novavax vaccine because I think that Novavax or the Oxford-AstraZeneca vaccines are probably going to be one of the two next vaccines that are presented to the FDA. Novavax is a protein-based vaccine, and is instead of injecting into the body the RNA or DNA that encodes the protein, the spike protein, instead what they have done is essentially created and purified and harvested the spike proteins themselves. And so the way they do that is they actually have engineered from the coronavirus, they've encoded and taken that segment of the spike protein, put it into something called a baculovirus. Baculoviruses are not infectious to people, but they are infectious to insects. And so this DNA virus infects moth cells. And so these moth cell cultures are used to then produce the spike proteins. And then the spike proteins are harvested, put into nanoparticles, and then combined with an adjuvant from a bark. And then that's what's injected into the body. And then again, taken up by antigen presenting cells and presented to helper T cells and B cells and all those sort of things. And so that's the Novavax vaccine. And again, I think that this is probably not too far away from presentation to the FDA. The last mechanism, there are other mechanisms that are being worked on, but the last one that is being commonly used around the world and not being tested here in the United States and not likely to be presented to the FDA, but these are the inactivated virus vaccines. And these are being used in some Chinese vaccines, some Indian vaccines, but they're used around the world and are being injected in a lot of people and are produced very cheaply, and which is part of the reason why they're being used in a lot of places around the world. And this is our oldest technology when it comes to vaccines. And so what they literally do is they inactivate the coronavirus, the SARS-CoV-2 virus, with a chemical. They take that, add an aluminum-based adjuvant, and then inject that into the body. And then the antigen presenting cells then chew that virus up and then present multiple different possible antigenic sites, but then presumably that's how the vaccine makes its immunity. So I want to, so aside from the inactivated virus ones, the other vaccines are actually in a lot of ways very, very similar to one another in the way they're ultimately trying to get the body to see the spike proteins. And so one of the thought experiments is, why is it that if all these vaccine candidates are meant to target the same protein on the surface of the SARS-CoV-2 virus, why should there be a lot of differences among the different vaccines' efficacy? And this information I'm going to share with you now is actually not widely reported in the press, but some of the biochemists and the molecular biology people are really fascinated by this topic, which is that the SARS-CoV-2 virus, actually the spike that sticks out actually has two shapes. And the two shapes, on the one on the left is the shape of the protein on the virus before it binds into the cell. And it turns out that conformation is actually pretty unstable. And as soon as it binds to the ACE2 receptor on the cell, it pops into the configuration on the right. And this right configuration then elongates, and then it allows the membranes to bind, and that's how the virus particle then fuses with the cell. And so two shapes of the same protein or set of proteins. And the recognition of why this became important is that a few years ago, if you remember, scientists have been trying to create a good vaccine against RSV for years. And so remember, RSV virus vaccines largely have not been that successful in preventing disease in infants. And a few years back, they discovered that there's a protein on the surface of RSV, much smaller than the coronavirus proteins. But it has a similar thing where it has a different shape depending on whether it's prefused or if it's post-fused. And the antibodies they were producing in the vaccines that they were using was against the post-fused shape of the protein. And so within the last 4 or 5 years, they've developed ways of locking that protein into the pre-fusion shape. And they've done it by essentially substituting a couple of amino acids, and that locking into that shape holds that position and allows the body to form antibodies against that much more effectively. And so when a few years back, they were trying to develop vaccines against the first SARS virus and against MERS, they used this locking mechanism to lock the proteins in a prefusion state, and they ultimately ended up with much better immunogenicity than they had gotten before. And when you look around the world at the vaccines that are using these different technologies, on the left here, the Moderna, the Pfizer, the Novavax, and the Johnson & Johnson vaccines all are using the shape, the prefusion spike shape here. And these other vaccines, including the Oxford-AstraZeneca vaccine, are using the post-fusion spike shape. And so does it matter? We don't really know for sure. There's a lot of guessing that's involved. But you can imagine that if your goal is to try to neutralize virus particles before they invade into cells rather than afterwards, then maybe you might see a better and more favorable response when you're using the prefusion configuration than the post-fusion configuration. So some really interesting science going on, really fascinating stuff. A lot of stuff that we're still guessing, we don't know, but that's just a quick update on how these vaccines work. I'm going to pause there because I can't see anyone. Any questions? Anything?
Just one in the chat right now. Why was the traditional inactivated virus approach of vaccine not an option / not working for COVID?
Yeah, it's a really good question. I don't actually know what were the reasons that certain vaccine candidates were chosen for Operation Warp Speed and other ones weren't. So quite honestly, I don't know. The other thing that turns out to be important as we talk about the variants, of course, is that these were all based on the original virus samples and the original genome sequences. And as these things have been changing, these are the things that we're facing and worrying about is the rapidly changing nature of the virus. And so it seems like one that is tried and true. So I don't know the answer. Cool. Let's talk about efficacy and how we define efficacy. This graph should be pretty familiar to most of you by now. This is the Kaplan-Meier plot from the New England Journal of Medicine article about the Moderna vaccine from December. And so these are the phase 3 trial results. And the Pfizer vaccine was almost identical. It was very, very similar. So we'll just focus on this one for now. And so if you look at this vaccine, so the arrows here are the 2 injection times, and they started measurement 2 weeks after the second injection. And you see that almost immediately you see a separation in the curves. And not only do you see a separation in terms of who's getting symptomatic COVID, this response is remarkably flat even after 4 months of observation in the vaccinated group. And they've given for you the incidence rates here. They've calculated these out for you, and they've calculated out the efficacy. And so when we're talking about vaccine efficacy, I've given you here on the screen the formulas in which statisticians calculate efficacy of vaccines. But I'm going to simplify it. The vaccine efficacy is essentially 1 minus the ratio of these events in the 2 arms of the study. Okay, so if efficacy is the ratio of the outcome rates between the vaccine and the unvaccinated groups, if we take the events that the Moderna study had, so 3.3 events per 1,000 years, the person years in the vaccinated versus 56 per 1,000 in the unvaccinated group, you can take that ratio, so 1 minus that ratio, and since it's per 1,000 person years, I've just moved the decimal place. And so what you end up with is 1 minus 0.058, and that gives you 94% efficacy. Okay, so again, it's the ratio of the vaccinated infections versus the ratio of the unvaccinated infections. That's the efficacy rate. So I'm going to ask people to unmute themselves and just say, how are you explaining when, if anyone were to ask, if a family member were to ask, a friend were to ask, or a patient were to ask, how would you explain what 94% efficacy means, knowing that that's how efficacy is defined? Does anyone have a way that they're describing what this means to people? Anyone? Bueller?
So I guess I'll just talk. So I more or less say that that number is generated from the amount of cases that you would expect to be occurring and the amount of protection that you're getting from the expected amount. So it's like we said, comparing the number of cases in the people that didn't get the vaccine to those that did. And those that did, there was 94.2% of the cases that would have otherwise been infected, they did not.
Yeah. Thank you for taking the risk and putting yourself out there, by the way. This is hard. This is really hard. I have seen well-meaning people in the popular press imply that 6% of people are going to get the disease even if you get the vaccine. And that is not at all what this ratio is telling people. And I want to be very, very clear when I talk to my patients that and the reason this is important is we talk about the Johnson & Johnson vaccine, because the top line vaccine efficacy for the Johnson & Johnson vaccine is 66%. And so people are saying, well, if it's 66%, that means my risk of getting infection is really high. That's not what this is saying. That's not what efficacy means here. So it's a really complicated statistical term here, and it has implications in things like the uptake and acceptance of different vaccine candidates. And so this is actually one of those cases where, and again, I don't have the answer exactly, but I have been playing around with how to communicate this for people who are interested. And this is one of those cases in which I actually take the time to not just share the percentage, but actually share the numbers. So you can actually, I've done this before, where I've actually said to people, if I took 1,000 people and vaccinated them and compared to them to 1,000 people who weren't vaccinated, around 3 of them might get symptomatic COVID versus around 55 or 56 people might get symptomatic COVID in the unvaccinated group. And I don't know if that's better. I think that by sharing both sets of numbers, I'm trying to communicate an important point. Another way in which I've been playing around and talking about the numbers, I've said to people, out of the roughly 75,000 people who got studied with the Pfizer and Moderna vaccines, a total of 21 people who got the vaccine got COVID, and only one of them was severe and didn't go to the hospital even. Out of the 75,000 people that they studied who didn't get the vaccine, 376 of those people got COVID. So again, I don't know if it's better, but I think that I want to be clear to people, and I want to make sure that they understand that this number doesn't mean a 6% chance of getting COVID, or for the Johnson & Johnson vaccine, that doesn't mean a 34% chance of getting COVID, because that would be a really high risk of getting COVID. We wouldn't want that for anyone. Any other comments about communicating efficacy? Because it's a really hard concept.
Can you repeat, Dr. Choe, one more time, how you explain the numbers to your line for patients using numbers?
Yeah, so I say to people, again, I either share these numbers here. I say, out of 1,000 people, you might expect only 3 people get COVID versus 56 people get COVID out of 1,000. Or I say out of the 75,000 people who were tested in the 2 vaccine trials, 21 people of those vaccinated got COVID and only 1 person got it severely, compared to 75,000 people who didn't get the vaccine, 376 got COVID. Yeah, so I don't know if any of it's better, but I think that it gives people a sense of scale, I think, because the background rate of infection in these studies wasn't anywhere close to 5 or 6%. It was just a couple of percent. The risk of infection among the vaccinated was 0.05%, right? Or yeah, so it was much, much lower than the people who were unvaccinated. Again, really hard concept. I don't pretend to have the answer about exactly how to do this, but I would say just to probe with your patients, if they understand, if they come to you and say, I want the Moderna one, I want the Pfizer one, I don't want the Johnson & Johnson one, I think it's worth perhaps probing about what they understand about what the efficacy numbers mean when we're talking about it.
Dr. Choe, can I ask a quick question?
Of course.
When you're So first of all, I really like that way you described it. I think people, patients, us, like everyone as a human, like I feel like our brains are better. I just have a much easier time understanding like absolute numbers versus like, you know, the statistical ratios, because it's like easier to see. But when you're talking about those numbers you just gave, was that, you're talking about the study of 75,000 people, is that for one specific vaccine, or is that something you use for all of them?
What I've done there, and I should have said it up front, is I've actually taken the Moderna and Pfizer vaccine studies and combined them together. I've done that. I've done that understanding that the two are very similar in terms of their ultimate findings. And I'm really trying to simplify it, because I'm trying to say to people like, and I've even had, I wonder if all of you have had this issue. I've even had people say, well, I want Pfizer because it was 95% versus Moderna, which was 94%. I mean, I've even had people say that sort of thing. So that number that I've given you here, so this 3.3 per thousand versus 56 per thousand, this is actually from the Moderna study. But if that 75,000 people, what I've done there is combined the results of the Moderna and Pfizer studies. So probably not cool. My statistician friends will just tell me that I'm not doing the right thing there, but I'm trying to simplify a little bit.
Okay.
The important number that the important number that was buried in there that I really want to highlight when I'm talking to people is that only one person got what they called severe infection. And that severe infection is not what we would consider severe. They still were at home and they didn't get hospitalized. No one died. No one actually required extensive hospitalization out of the people who were vaccinated. That's compared to a lot of people in the unvaccinated group over this period of time who were hospitalized. So I think that's the bottom line.
That was 73,000 people in the study. One person got severe COVID.
Yes. It's amazing, right? It's totally amazing. It's an amazing feat of science that we got here in less than a year.
Hey, Dr. Choe, this is Chinonso. Yeah, I think this is a really interesting point. So basically, is the efficacy basically describing the relative risk reduction?
Yep.
So basically, people who get the vaccine are 94% less likely than people who don't get the vaccine. And so like the relative risk reduction, you know, the greater the baseline risk of the population, then the greater the absolute risk reduction. So it doesn't necessarily translate to absolute risk reduction.
Yes,
Right.
that's a really important point, and that's really helpful. So if the background rate, let's say that we're in total crisis mode, like early days of the pandemic, and let's say that we're in a situation where 10% of the people were getting sick. And so if you talk about the risk reduction in that case, if you use the same relative risk reduction, you're going to see that the actual numbers of people who are impacted here are much greater because the background risks are greater, right? So these are really important. Chinon, so you make a really important point that rate here, this 56 per thousand, is what was going on at the time of the Moderna vaccine, right? And so that number might be higher or lower depending on what's going on in the community. If you figure this out, you can do the math and figure out what that number would be depending on what this rate is. So this, in short, this becomes much, much more important to talk about this and push this as the rates go up or down, depending on what's going on in the community. Okay, I'm gonna move on here, and if there are more questions, we can come back to this. But remember, you know, so that's the top line efficacy number, 94%, right? But remember that when we're talking about efficacy of clinical vaccines, we're really also important. We're also really wanting to make sure that clinically important subgroups are included, right? And so clinically important subgroups that were predefined in the study are the ones that we probably are really worried about. So how did the vaccine do in the old versus the young? How did the vaccine do in groups with medical comorbidities? What about in racial and ethnically diverse communities? And so luckily we see here, they've graphically depicted here and shown you with these numbers, is that this vaccine does really well in all these predefined groups, maybe slightly less well in the older groups, but still very, very well. And so that's also very important as we think about communicating these risks and benefits to patients. Okay. I'm going to shift gears and talk about the Janssen or Johnson & Johnson vaccine, because again, I think most of you are probably pretty familiar, or at least have some working familiarity with the mRNA vaccines, but this is brand new. And in full disclosure, before preparing for this, I didn't actually know a lot of these numbers. I saw what was on the news. I'm going to share these references at the end, but this is a really helpful spreadsheet that's being actively updated on a regular basis from the American Society of Health System Pharmacists. The link is there on the right-hand side there, but again, I'll put it at the end. This group is actively updating information regarding current vaccines and also candidate vaccines. The actual spreadsheet is much larger than what I've shown you here. And on the spreadsheet, they actually have information about what things are being updated, what's being used to try to qualify them for FDA approval and those sorts of things. And so I want to actually pause and say it's probably worth reminding people how the FDA approves vaccines in the United States. And so all three of these vaccines, the Moderna, Pfizer, and the Johnson & Johnson one, have been approved under a category called emergency use authorization. And so in order to meet the criteria for full authorization, they actually have to have a really high standard of proof at the FDA, and that includes lengthy follow-up data. And in the middle of a pandemic, when everyone's dying, you obviously don't want to wait for the full lengthy follow-up because waiting's too dangerous. And so they have this category, this emergency use authorization. In order to meet that, they still need to meet the same burden of proof of efficacy, and they need to be proving and watching as best as possible side effects and adverse events. And so I've heard this a lot from patients, and you probably all have too, that it was too much, too fast, too soon. And that how can you know whether it works in such a short period of time? In fact, the proof of the efficacy is the same regardless of whether it's full authorization or emergency use authorization. It's all that follow-up stuff that's different. In the United States, well, let's go to a place like Britain. Most other places in the world, instead of what they do is they essentially take the data and how it's presented to them from the drug manufacturer or the vaccine manufacturer, and they read that brief that's given to them, and they essentially approve it or don't approve it based on what the drug company presents to them. And the FDA, and this dates all the way back to many, many years ago, back to the days of thalidomide and all sorts of bad drugs. The FDA actually insists on getting the raw data. And they insist, not only do you present us the information of how you want to package it, but we want to see all the data that you have, and you need to give it to us so our own scientists can look at this. And so if you might wonder, why is it that the Oxford-AstraZeneca vaccine has been approved for so long in Britain and England, and we haven't approved it here? The Oxford-AstraZeneca vaccine hasn't yet applied for emergency use authorization here in the United States because there's been a lot of delays in getting the data that the FDA has been wanting, and the FDA has been asking for, and they haven't been able to provide it to them. And so for vaccines, the FDA leans heavily on what's called the Advisory Council of Immunization Practices, the ACIP. It's a committee of the CDC that includes academic researchers and CDC scientists. And they regularly meet and review vaccine data. If you've ever wondered who those crazy people are that keep changing the Novavax versus Prevnar and what order they're giving it to you, it's the ACIP. That group is regularly meeting on an ongoing basis and update vaccine recommendations. So the ACIP is going through the data that the FDA provides to them. They do the initial analysis of the data, and they essentially vote on those, whether to authorize it. And the FDA technically has to then review it and accept it or not, but for the most part, they're taking those ACIP recommendations and accepting them as they are. And so if the ACIP approves it, then largely it's getting approved for emergency use authorization. And so for the Johnson & Johnson vaccine, we don't actually have a lot of data that's been shared publicly in the larger population, right? So we don't actually have a New England Journal of Medicine article yet, or a JAMA article, or Lancet article. For the Johnson & Johnson data, what we do have is the advisory committee ACIP notes, and they've published their notes in their summary of those data. Can you hear me? Did I lose audible?
I can still hear you.
Okay. Yeah. So we have essentially in the Morbidity and Mortality Weekly report from 2 weeks ago, we have their meeting notes. And so what I've done, what I'm going to show you is sort of a summarized version of their meeting notes that is their review of the phase 3 clinical trial data. And hopefully they'll publish this in one of our medical journals so we can all read the data directly. So here's the top line efficacy information. So vaccine efficacy, this is directly quoting from their paper. Vaccine efficacy against symptomatic laboratory confirmed COVID-19 was 66.3% after 2 weeks after the injection. The Johnson & Johnson trial defined their outcome a little bit differently than the Moderna or Pfizer vaccine trials. They're a little more strict about how they define this, but for the most part, a lot of it kind of overlaps the Moderna and Pfizer definitions of what they were looking for. Importantly, like we said for subgroups, the effect size was pretty similar and consistent by age, sex, and race, and whether or not someone had underlying comorbid conditions that were of interest. This number here, the 66%, I've already alluded to it, but I think that it's really important to sort of pause here and sort of untangle this for patients because, again, I don't know, maybe you can all sort of raise your hands or whatever, but I've already had lots of people say that they don't want this vaccine because of the 66 versus 95%. And in the news a couple weeks ago, you probably saw this piece that, you know, I think it was the mayor of Detroit or some other city towards the east that said that they didn't want, or maybe it was Cincinnati, they didn't want the Johnson & Johnson vaccine because it was less efficacious because of 66%. So we've already said that this doesn't mean that 44% of people will get COVID-19, and we're going to come back to it. But I think that this is, if anything, we're almost sort of a victim of the high success rate in the mRNA vaccine efficacy, that we have a little bit of a marketing issue. And I won't get into it here, but I know that Hillary and Becca are going to talk about vaccine equity issues. I think that there are perceived equity issues about whether someone is offered the Johnson & Johnson vaccine versus the Pfizer and Moderna vaccines. Importantly, this is the number that I think that a lot of public health officials are pointing to, and I think that you should probably point to when talking to your patients about this. It is highly protective against COVID-19 hospitalizations. So the event, again, these are 20,000 people that were studied in this. There were 29 people in the placebo group out of those 20,000 people studied that got COVID-19 versus only 2 in the vaccine group that got it and were hospitalized. After 4 weeks, 16 people in the placebo group went on and got hospitalized for COVID-19 and none in the vaccine group. So, you know, again, 93 and 100% efficacy at protecting against hospitalization for COVID-19, which is, of course, what we really want. And it is protective, we think, against death from COVID-19. None of the 7 COVID-associated deaths happened in the vaccinated group. And this part didn't make a lot of the news, but I think this is really exciting. This is really important to say that it probably also protects against asymptomatic infection. They didn't test everyone, but in a subset of people, they actually did serologic testing after, you know, about 10 weeks, where they did serology testing in all these in a bunch of these volunteers. And of that group, they were testing serologies against a different protein, not the spike protein. And they found that about 3% of the placebo group tested positive who hadn't known that they were infected. So in other words, asymptomatic infection at some point during the past 70 days, versus less than 1% of the vaccinated group. So this is the thing that we've all been wanting. We've all wanted proof that not only is it preventing symptomatic disease, which is what's super important, but is it also preventing asymptomatic disease? And this seems to be probably true for the Johnson & Johnson vaccine. So again, a really important point about this. This part is important, and this part I think we've got our eye on as we talk about the Johnson & Johnson vaccine. So the efficacy varied by region. It was a multinational study. It was done in the United States, parts of Latin America, and parts of South Africa. And you can see here the vaccine efficacy rate drops depending on which region you're looking at. And so probably your next question, and naturally, is what about the variants that were going on in those places? And so they looked and they found that in the Latin American portion of the studies, they found that the P2 variant was very prevalent, particularly among the volunteers from Brazil. And the B1351 variant, the variant that originated out of South Africa, that's really worrisome to a lot of scientists, that was almost 95% of the circulating SARS-CoV-2 variants in South Africa. So there's at least a pretty strong signal that while there's protective effects, that it's diminished depending on which variant you're looking at. If you're thinking about this statistically, this 52% efficacy still reached statistical significance. So it was still considered statistically significant. This part I think is really exciting and really important. And this is the part that I've actually been using in talking to my patients, which is, I won't ask you to show your hands, but how many of you had severe reactions to the mRNA vaccines? So local reactions to the mRNA vaccines, if we just take the Moderna one here, we can see that like almost 90% of people had local reactions to the second dose of the Moderna vaccine. And that's compared to 60% of the young and 35% of the older group of the Johnson & Johnson people. So already a lower rate of local reactions. For systemic reactions, we see that, again, 80% of people had systemic reactions after dose 2 of the Moderna vaccine, and lower rates of systemic reaction among the Johnson & Johnson. And this part is huge, which is that if you look at those more severe reactions, those we call grade 3, 16% of people in the Moderna vaccinated groups got greater than or equal to grade 3 systemic reactions. So higher numbers are worse. The Johnson & Johnson reactions, it was 2% and 1%. So much, much lower rates of severe systemic reactions. And also important, and probably can't be overstated, is that it's one injection and obviously easier to administer on a large population basis, but also for my patients who are extremely needle phobic, this is a really key point for them, is that it's the one injection rather than 2 injections. So to summarize, and I hope you take away these points when thinking about this and talking to your patients, the Johnson & Johnson vaccine, so the efficacy against symptomatic disease was reported as lower than for the Pfizer Moderna vaccine, 66% and 94%. I think there's a lot of confounding issues. The study is multinational, included locations, and included times where different variants were more prevalent. And so we don't actually have necessarily the same head-to-head comparison. So that's important to say. This vaccine, though, just as the mRNA vaccines are, is highly effective at preventing hospitalizations, greater than 93% efficacy, and death, 100%. These absolute numbers were small in these 2 groups, but overall the effect size was probably pretty similar to the mRNA vaccine. So yes, the head-to-head comparison, the top line efficacy looks lower, but the part that's super important, the ones that we really care about, absolutely the same as the mRNA vaccines. And that it's easier to administer, fewer side effects, 1 shot versus 2, and much, much lower rates of severe side effects. I'm going to change subjects in a second, but I want to pause because I saw some things flashing through the chat very quickly that I didn't get a chance to read. And so Courtney, if you can summarize some of the questions.
Yeah, I'm happy to help. There was a request from earlier from Becca about possibly putting together some cheat sheets for residents to have with the numbers that you sort of, and
Sure.
the link that you use for patients to be sent out afterwards.
Sure, we'll do.
And then a question about, are asymptomatic patients still able to shed virus despite being vaccinated? What do we know about that?
Don't know.
And then another question, what's considered greater than a grade 3 systemic reaction?
Oh, really good question. I should know this. How many of you, any of you used that CDC, what the COVID symptoms tracker thing? I can't remember on there, but if you go through there, there's a part where it asks for severity. And I think that some of the questions that are on there about like, did you have to lose work or change your things because of it? I think that's part of the stuff that goes into the severe reaction, but really good question. I should look that up.
And then a question about, how patients are asking about how covered people are after their first shot.
Oh, good question. I try not to. So there's a lot of questions about this, and this came up when different countries were talking about expanding or extending. Britain has said that people can extend that for very long periods of time. That spreadsheet that I showed you actually goes through and talks through this, but let me give you some rough numbers. So the Moderna and the Pfizer mRNA vaccinations, after the first vaccination, it's somewhere on the order of about 55% for the Pfizer vaccine and about 70 or 80% for the Moderna vaccine. I don't know how long after that vaccine. I don't think it's immediately. I think it's something like 2 weeks after that vaccine. I don't know that same data for any of the other candidates, but obviously the Johnson & Johnson one's the one vaccine. So there is some coverage. I'm really trying to urge people to pay attention to those, say 2 weeks, keep masked and all that sort of stuff. But there is early protection. We saw the curves for the Pfizer study start to diverge after about day 11. So that tells you that there's probably some protective antibody effects starting from day 11. Yeah, COVID arm, thank you, Shiv. COVID arm, if you didn't see this, this is a real thing. It seems to be more prevalent in the Moderna group. It's largely not reported as much in the Pfizer group. The COVID arm issue is that there's a late stage reaction that happens almost predictably on day 8. It seems to be a histamine mediated reaction where someone gets a very large indurated wheal or something like a large spot on their arm where they got the injection. We've been telling people that antihistamines, over-the-counter antihistamines, seem to be effective at it. So it seems to be a histamine mediated response, but beyond that, I think still under investigation. AstraZeneca safety controversy. We're going to talk about that. We're going to talk a lot about that. Okay. Did I cover all the questions, Courtney?
Yep, you got it.
Okay, great. Let's talk about, I want to talk about the Novavax and the Oxford-AstraZeneca vaccines. And the reason I want to talk about these is because I think these are probably going to be the next 2 vaccines that are presented to the FDA for emergency use authorization. So what I'm presenting to you for each of these is really sort of interim data and incomplete data, but this is me calling through the literature, trying to find and looking through a variety of resources, trying to find as much data about these as we can. So Novavax, again, this is the protein-based vaccine with the adjuvant. So we have interim phase 3 trial data from the United Kingdom. And so in the UK, let me actually, before I get into this data that I'm sharing with you here is from a press release from Novavax. So take this with a huge chunk of salt. So they're going to present this in the most favorable light, but let me just share with you what they presented. So out of the 15,000 participants in the UK, they saw about an 89% efficacy rate. So 56 cases in placebo, 6 in the vaccinated group. And like the other vaccine candidates, there were no severe cases among the vaccinated group. Importantly, as this was Britain, it showed an 86% efficacy in the B117 variant. So that's good, that's important. And we care a lot about that because that's going to become much more prevalent here in the United States. A little less favorable and a little more worrisome, but still generally positive is that we have interim phase 2b trial data from South Africa. And so data out of South Africa, what they saw was that the efficacy against symptomatic disease was down to 49%. I don't have the numbers. They didn't calculate a 95% confidence interval for us. Less effective. They tried to put a positive spin on it. This was, they split out then the data between those with and without HIV and said that the efficacy was better among those without HIV. And they sequenced not all the cases, but a bunch of the cases. And of those, greater than 90% were the more worrisome B1351 variant that we have been seeing in South Africa, and we're watching this very carefully. So we're gonna have to follow this pretty carefully. We'll have to see what the final data show from South Africa in particular. I wanna really look at their 95% confidence intervals once they present them to us. But I think this is something to keep an eye on. They've completed enrollment and they are just about done with interim follow-up data from the trial the phase 3 trial that has been going on here in the United States and Mexico. And you may know that Seattle was one of the recruitment sites. UW was one of the recruitment sites for the study. And so they had a press release a week ago. It was all over the news. It was all over the national news that said, this press release said that the immediate data that they had available showed 96% efficacy. So that's huge, right? That's even better than what was reported for the Pfizer and Moderna vaccines. I went back last night and looked for those press releases, and they've been pulled from their sites, from the Novavax site, which makes me worried that somehow maybe those numbers weren't right and that they've relooked at them. So I'm a little worried about what that means, that they've pulled it from their site. But I expect that they're gonna submit this for emergency use authorization within the next couple of weeks to months. Let's talk about the Oxford-AstraZeneca studies and the data behind this. Ryan says, I don't understand why there's not an expectation that this primary data is all public. Yeah, Ryan, I think that the issue is that they will have to present it to the FDA. I think that the press releases that they're doing is not meant for us as physicians. It's meant for investors, right? So again, huge grain of salt about all this information. We'll have to see what they officially present. Let's talk about the Oxford-AstraZeneca studies. So they have been doing some studies here in the US. We don't have that data yet, but they have presented, and these are the data that they used to get approved in Europe. So they did a pooled analysis where they took studies that were doing in multiple parts of the world, including obviously in Europe, and they presented for approval an amalgam of pooled data from multiple different trials and countries. And these were phase 3 studies, these were phase 2 studies, some of them were even from phase 1 and 2 studies. So these studies are really a mishmash of a lot of data. The outcomes that they were looking at, the clinical outcomes, were defined pretty broadly, differently from some of the other studies. And what they found in when they looked at these 11,000, almost 12,000 patients, they found 30 cases in the vaccinated group, 101 in the control group. So they calculated an efficacy rate of 70% in preventing symptomatic disease. Like all these other vaccines, very effective at preventing hospitalizations. None of the hospitalized patients were among the vaccinated group. Parts of this that have given a lot of public health officials here in the States a reason for pause is that they kind of did sloppy science. I'm editorializing a little bit. Some history here. AstraZeneca, so Oxford, the researchers at Oxford had this vaccine developed and ready to go, and they needed a company to partner with, and they partnered with AstraZeneca after shopping it around. AstraZeneca is very good at producing medications. They've done a lot of that. This is actually the first large-scale vaccine they've produced. And so there are a lot of issues about production that happen immediately and early on. One of the things that happen is that a small group of people in the initial studies accidentally got the wrong dose of their first injection. So they got like sort of half the dose of vaccine in the first injection and got a standard dose in the second one. And what they found completely by accident is that this group that got the smaller injection first had better efficacy than the group that got the standard dose injection for both injections. And a lot of these data, so like if you look here, 90% in one versus 60%, 2% efficacy in the group that got the study in the way it was intended. So some of the benefit is being driven by the smaller group that had better efficacy. Most of the volunteers were white. This was, a lot of this was done in Europe. And they had very, very limited numbers of older patients. Very few patients in the proportion of the patients who were studied older than the age of 55. And so some countries aren't even giving this to those older because they rightfully point out that we don't have a whole lot of data of effectiveness in older patients. There's a US phase 3 trial that's just been completed. It's about 32,000 patients that have been enrolled and studied in the United States. And the expectation is that sometime in the next probably 4 to 6 weeks, they're going to present these data to the FDA. There's a history here that's important to understand, which is that the FDA is kind of pissed at the companies who are producing this because back in December, there was a pause in the studies because there were a couple cases of transverse myelitis that were discovered. And the manufacturer or the people studying this paused the trial, but they didn't tell the FDA. And they didn't tell the FDA that they'd paused it. They discovered it in one of these press releases that went out, or in investor call, I think is what it was. And so the FDA stopped things and said, you need to give us your data. And we need to look at this ourselves because we want to look at this a little bit more completely and understand what's happening here and see if we agree that this is not a concerning issue. And they had 3 weeks to produce that data. And I believe that it took them almost 2 to 3 months for them to produce that information back to them. So there was a long pause during which time the FDA never got their data. And so there's been a history here and there's been an issue, and that they're going to scrutinize these data really, really carefully. And that doesn't even take into account some of the stuff that's going on currently with the Oxford-AstraZeneca vaccine. I'm going to talk about that in a second, but let me actually pause here and see if there are questions that I've missed.
There's nothing so far in the chat.
Great. Okay. I know I'm going a little bit long here. We'll take a break in a bit, but let me actually whip through some late breaking updates. And when I say late breaking, these are within the last 2 weeks. This came out on Tuesday. This study is in the New England Journal of Medicine this week. This study came out 3 days ago, reporting on the results of the study of the Oxford-AstraZeneca vaccine in just over 2,000 HIV negative adults in South Africa. And what they found is, not surprisingly, there was huge numbers of the B.1.351 variant. In these 2,000 or so HIV negative adults, they got placebo. It was a 1-to-1 randomization, placebo or the vaccine. Mild to moderate COVID developed in 3% of the placebo group and 2.5% of the vaccine group. When they calculated this out, when they plugged the numbers, they calculated out an efficacy rate of 22%. And you can see here that it's not significant. The 95% confidence interval, 60%. You know, small numbers, but if in essence, there was no benefit given by the vaccination. Among the 42 people who got COVID-19, 39 of those were the B.1.351 variant. And when they calculated it out to that group specifically, the efficacy was 10%. They went back and looked at some neutralization assays. So they took the serum, they looked to see how well the vaccine was, the antibodies from that person's serum was working against a preset amount of virus. And although the numbers were small, the placebo group that had shown previous infection performed better at neutralizing the virus than those that got vaccinated. Only 4 people in placebo group, so don't necessarily hang your hat on that. But all of that is to say it's not looking good for this vaccine for the variant, the B.1.351 variant. if you're a graphic person, like to see this, the Kaplan-Meier curve here looks terrible. And so the salmon shade here is the placebo group here. This is the placebo group in infections that developed. The salmon colored shading here is the 95% confidence interval for that curve. The blue here is the vaccinated group. And if anything, you see that there's actually more events here among the vaccinated group. And here's the shade of the 95% confidence interval. And you see that they overlap, so not statistically significant benefit. So my takeaway on this is that even though it's small, 2,000 people, it looks like it was very well done. The study was pretty high quality, hugely worrisome that there's any benefit at all for the Oxford-AstraZeneca vaccine for at least one of the scary escape variants that we're worried about and seeing in many parts of the world. So that's worrisome. That's from 3 days ago. Even more recently and ongoing is this issue about thrombotic events. And as of this morning, I was checking my news and clicking on refresh. The EU health commissioner, whoever the czar is for making these decisions about vaccines, was supposed to announce today a decision about whether to proceed on with the AstraZeneca vaccinations. More than 20 countries as of yesterday morning had paused vaccinations, more than 20 countries. And this is huge, right? Many op-eds have pointed out, and probably they're going to proceed on and go on with vaccinations. Many people have pointed out that the rate overall of thrombotic events is not higher than what's expected in large populations generally. And so if you look at the incidence ratios between the vaccinated and unvaccinated groups, it doesn't look like there's more of these events among the vaccinated group. What's a little bit scary about this, so the news about this and the reporting about this is very, very sparse. And I had to dig through pretty deeply and to try to find some of the stuff. That news article that I showed you a page ago, this is from a pretty reputable site. There's some worrisome features that have given people pause. The regulators in each of these individual countries understand how big a deal it is to pause vaccinations in their countries. And so one of the things that a lot of people have been wondering about is that, well, why are they stopping? How worrisome is this? What's worrisome is some of the descriptions of what's happening, the clinical features of some of the clotting events that are described. So what people have been able to pull together is that it looks like at least 13 patients have had these events. And these are people who are previously healthy, no other health issues that they know of, between the ages of 20 to 50. At least 7 of them have died. And what's reported, again, this is not a scientific paper, this is through pulling together a lot of different sources. What's been reported is that those cases involved widespread clots and thrombocytopenia and hemorrhage, internal hemorrhage events. And 7 reports of cerebral venous thrombosis. And so people are wondering if what's being seen here is some sort of DIC kind of picture. Is it like an HUS kind of picture? What exactly is going on? It doesn't sound like it's just like a DVT, like a spontaneous DVT or a PE or something like that. It sounds like these worrisome events are much more widespread through the body than a single event. And so this is not just a clot that they're looking at. This is worry about, is there something else going on? There's some hand waving, like is somehow the complement cascade being activated? Is there some DIC kind of picture? So no one exactly knows. And it's also important to note that there are some cases of some thrombocytopenia and thrombotic kind of events happening among the mRNA vaccines that are currently under investigation. The FDA is investigating what seem to be about 30 or 35 cases of ITP-like events. But these seem to be without these widespread clots. So is there something going on about this vaccine? Is it activating something going on in the immune system? Jennifer Dunlap, I just got a news alert. Not linked, benefits outweigh risks. So that's not a surprise. What I think is really important and really the big issue here is that if you look through and you follow the news, if you look at news sources in other parts of the world, in other countries where this has been paused, this has probably caused what might be irreparable harm to the acceptance of this vaccine in Europe. And I'm being a little cynical here, but I think the other news story that I came across is that the US is talking to Canada now that we have a stockpile of about 30 million doses of this vaccine. And we've begun conversations with Canada that we might give up some of our supply of the reserved AstraZeneca vaccine for their use. So who knows? I think it's worrisome. I think it's scary. I think that we need to keep our eye on this. I think that the FDA is going to be looking very, very cautiously at this. I think this is, I'm not sure how they'll accept the Oxford-AstraZeneca application. I think that there's a lot of positive features about it, but I think that there's been a lot of issues with it. And given how we're starting to do better on some of the other vaccine sources, it's not clear to me whether we'll see this or whether the FDA is going to force AstraZeneca to come up with different data to watch. So keep an eye on that. All right, I'm going to finish with one positive thing, and I want a positive study, and I really want to finish on a high note here. Let's end on a positive note. This is from last week, clinical infectious diseases. So we've been waiting for a long, long time now, right? We've been talking on and on about whether COVID-19 vaccines don't just reduce symptomatic disease, but do they actually reduce asymptomatic disease? And by extension, does that reduction of asymptomatic disease mean that we can reduce the risk of asymptomatic spread? We have some pretty suggestive ecologic data. Right? There's been some pretty dramatic reductions in places like Israel, where they have a single payer health system. They've vaccinated many Israeli citizens. There are a lot of issues about bias, a lot of issues about confounding. What's going on might be the reductions in infection rates might not just be their vaccinations. So there's a lot of things going on. There's also some small pilot data that I mentioned, like in the Johnson & Johnson vaccinated groups and in the AstraZeneca and Moderna vaccine trials. So there have been some suggestions of this, but we've been really looking and waiting for real world proof of this. And last Monday, a week ago, this paper appeared in this early release form in the Clinical Infectious Diseases. And this, I love this study because it's so beautifully elegant and simple. So what the study did is these authors took people in the Mayo Clinic system who were getting elective procedures. And at Mayo, just like here, all the people who are getting elective procedures all have to get screening COVID tests. So they took all these people, the 48,000 people between mid-December and mid-February who were getting elective procedures at Mayo, and took all the data of their screening tests and looked at the results. The 3,000 people who were screened who had had at least one COVID vaccination were looked at separately from the 45,000 people who hadn't yet gotten COVID vaccination. So of those 3,000 people who'd gotten vaccinated, there were 42 positive tests, 1.4%. So in other words, these people unexpectedly, they didn't know that they had COVID. 1.4% of those people who had gotten at least one vaccine were positive. And of these people who were not vaccinated, 3.2% of them had positive tests. So when you do the math here, there's a relative risk of 0.44. And when they looked at those people who had been at least 10 days after the first vaccine dose, the relative risk was 0.2. So what they were looking at was that there was almost an 80% risk reduction in asymptomatic disease seen when they compared the cohorts of those that had been vaccinated versus those who weren't. Were those PCR tests? So yes, those were PCR tests, not serologic tests. So this is what we've been waiting for, right? This is the information, and I don't think that it's any coincidence that the CDC updated their guidelines about the fact that those that are vaccinated can start removing their masks in very, very specific situations. So I'm going to stop there, say that despite all this complicated data around vaccinations and hard to wrangle all that information, we really are seeing the light at the end of the tunnel, thanks to the vaccines. And so I think we'll have to take a break, but when we come back, I know that Hillary and Becca will want to talk about how we take this information, how we communicate it with patients and help them come to understanding about whether to get vaccinated or not. And I'll share these slides later. These can be sent around. The top two references there, I haven't given you all the references I've included. They're on the individual slides. But these top two references here are ones that I think that if you want to bookmark two places, the top one is that spreadsheet that I was talking about that has information that's updated regularly about the vaccines from the American Society of Health System Pharmacists. The second one is a collaboration between CDC and IDSA. It's the COVID-19 Real-Time Learning Network. This includes regularly updated information where they're culling through the pre-publication clinical trial data, they're updating it and including on their website information about vaccinations and all things COVID. So I don't know if there are any last questions. Thanks for letting me share all this.
Awesome. Thank you so much, John. That was so helpful and comprehensive. Let's give everyone a 10-minute break. So back at 10:25 for Hillary to take over. And then if there are questions, I guess, in the interim, we'll be here.
There's a request for me to include the links in the chat. I would be happy to do so. I'll do that in a moment.
Hey, Dr. Choe, this is a little bit of an off-topic question, but has there been any data about the schedule for children to get vaccinated at all?
Yeah, do you have kids?
I do not, but I get asked from my sister.
Yeah, exciting news. So the Pfizer is approved down to 16, Moderna down to 18, Johnson & Johnson down to 18. Pfizer has completed, I believe, I think both Pfizer and Moderna have completed enrollment. They're still collecting data on the 12 to 16-year-old groups. Moderna this week announced that they are starting recruitment on 12 down to, I believe, 1 year of age or maybe longer. So they're actually starting to recruit from those groups. There's a nice New York Times article about the challenges of recruiting kids and some of the ethical considerations, and it's a nice read. It's challenging. Among the things I read in that never occurred to me is that they are discouraging participants from including that on social media because they're worried about them being targets of sort of nasty things on social media.
Mm-hmm.
You know, all sorts of things that teens love to do, share on social media, they're discouraging those kind of things. So there's a lot of ethical issues. But the expectation is that the 12 to 16-year-olds will have data starting probably around midsummer. We'll have some initial data. And, oh, thanks, Kim, you're so fast. And then the younger group probably by fall. Now, the turnaround time is typically about 8 weeks for the ACIP and FDA to make decisions. They're promising to fast track all these decisions because obviously everyone is looking to the fall in schools.
Awesome.
Yeah, that's what I was just gonna say.
Sorry, go ahead.